Good morning to everyone. Myself P. Aparna yadav, MSc first year in SK University, Anantapur. Thank you for giving this golden opportunity, Dr. D. Muralidhar Rao sir, Assistant Professor, Department of Biotechnology in SK University, Anantapur. Today we are discussing about DNA footprinting. What is footprinting? Footprinting shows where a target lies on DNA and which bases are involved in protein binding. Three methods are very popular: DNA footprinting, dimethyl sulfate footprinting, hydroxyl radical footprinting. DNA footprinting. Introduction. DNA footprinting is a method of investigating the sequence specific of DNA binding protein in vitro. This technique can be used to study the protein DNA interactions both outside and within the cell. History. In 1978, David Gullers and Albert Smith developed the DNA footprinting technique to study the binding specificity of the lac repressor protein. It was originally a modification of the Maxam and Gilbert chemical sequencing technique. First, we should take the genomic DNA and amplify it by PCR and form the labeled template DNA containing the protein binding site and the protein of interest. Here we can see that proteins are attached to the labeled template DNA. Third step, we can cleave the DNA. Fourth step, the cleaved DNA is on polyacryl amide gel. We can see that protected footprint. DNA footprinting is an in vitro molecular technique used to identify the protein binding region on a DNA molecule. This technique is mainly used to identify the transcription factors that bind to the promoter, enhancer or silencer region of a gene to regulate its expression. The regulation of a transcript ion of a gene can be studied using this method. This technique is also called as DNase 1 footprinting. Thousands of proteins are interacting with DNA in the nucleus for regulating activities like replicating, transcription, translation, etc. Transcription is a process where the DNA is converted into RNA in the cell nucleus. Initiation of transcript ion takes place when the enzyme RNA polymerase binds to a gene sequence known as promoter sequence. DNA footprinting can be used to identify RNA polymerase interacting DNA sequence. Principle. In this technique, nucleases like DNase1 is used which will degrade the DNA molecule. Nucleases cannot degrade the DNA if it is bonded by a protein. Thus, that region is protected from degradation by nucleases. This protected DNA region is called footprint. Procedure. First, DNA fragment containing protein binding sequence is extracted, amplified and labeled at one end of the double helix using polymerase chain reaction technique. Label DNA fragments with DNA binding protein and cleavage. Mixed it in a test tube. In another test tube, labeled DNA fragments are mixed with cleavage agents without DNA binding protein. This is used as a standard to compare the result. Cleavage agents cut the DNA fragment present in both the test tubes, but no cuts are made at the specific region of DNA. Where proteins are bound, proteins has protected the DNA binding site from cleavage agent. DNA fragments are separated by polyacryl amide gel electrophoresis and visualized using autoradiograph. When compared with the standard, missing band indicates the protein binding specific DNA sequence. Cleavage agents are used in DNA footprinting or DNase1. DNase1 is a double-stranded endonuclease enzyme that cleaves the phosphodiester bond present in the DNA. The enzyme action can be controlled by EDTA solution. The enzyme acts on DNA structure and sequence specific, resulting in an uneven ladder. This can also affect the precision of predicting protein specific DNA sequence. Hydroxyl radicals. Hydroxyl radicals are produced from a reaction where a iron salt is reduced with hydrogen peroxide to form the free hydroxyl molecule. The free hydroxyl molecule cleaves the DNA fragment. Hydroxyl radicals are independent of DNA sequence for their action, therefore form evenly distributed ladder, but the reaction rates of these radicals are very low. Therefore, it requires more time to cleave the DNA fragments. Ultraviolet radiation. Ultraviolet radiations are used to excite the nucleic acid, and this may lead to damaged DNA fragments. Applications. DNA footprinting can be used to determine the sequence-specific DNA binding protein site to study the DNA protein interaction. Transcriptional regulations can be studied to identify the functional genes present in the large genome of human. Researchers often use this technique to identify whether a particular protein can activate or inhibit transcription. Thank you.